For our next session, we have the distinct honor of having Dr. Anna Greka. And as Richard has mentioned, Anna is associated with MIT and Harvard Brigham. She's been a pioneer on behalf of those with rare disease. We've had an opportunity to have her at a number of different international formats, and she's compelling. And let me tell you what the distinctive is, Dr. Greka, from a patient standpoint. You want researchers and doctors who understand what the burdens of disease are in a practical way. And they take those experiences they've heard from patients and it fuels their work and their curiosity. And Dr. Greka, hats off to you because you've never lost the sense of the patients in any of your work. So why don't we go ahead and turn the floor over to you? We're always excited to hear from you, and especially the important work that you're doing and how we can support you. So go right ahead.
Wonderful. Thank you, Paul and Richard and Sharon and everyone at the Rare Kidney Disease Foundation. Some of you may have heard me speak yesterday as well, and so some of this may be repetition, but I actually was wondering about maybe making this more interactive. If there's questions that people want answers to, I can try to do my best to answer them. I can also briefly go over the points that I described yesterday so that everybody is on the same page. I just wanted to say that, it's a distinct and special honor for me to be with all of you today. And I think it's just remarkable, like the heartiest congratulations, because the Rare Kidney Disease Foundation has come so far. I mean, this is such an incredibly well organized virtual summit. Kudos to everyone who's worked to make this possible. I just think that, this organization started out with me basically saying to Richard and a small number of patients that, you have to get organized, your advocacy is going to be important as we develop treatments and try to move them toward the clinic. And of course, Paul and Richard know this very well, how important advocacy is. And so, kudos, like really, you have done such an incredible job organizing, and I know that there's more to do, but this is just a tremendous effort, and it will really pay off in the future because this kind of advocacy is very effective. Organizations like the FDA and the EMA in Europe, the European Medicines Association, loves to hear from patients, and it's most impactful if you tell your stories. Of course, we as the physicians and the scientists, try to make the best case possible for whatever treatments we're trying to push forward toward the clinic. But really, the patient voice is very, very important. And it's absolutely critical in the process of developing those drugs. That's why I wish for all of you to be our partners on this long and winding road, admittedly, until we can get to therapies. But we are, well on our way and very committed to making all of this happen. So again, I'm just honored and incredibly grateful and proud, if I can say, for the fact that you as patients have organized so wonderfully to advocate for these diseases. So it makes everything so much better. So I guess I will go through a few short slides like I did yesterday, but I really just want to open it up for questions as well. I do want to address one question that was just in the chat about iptacopan or Fabhalta. This is the new medicine made by Novartis, as it would be, but it is not for these diseases, and it's not expected to help with these diseases. It's approved for C3 glomerulonephritis and for IgA nephropathy, as well as in children with paroxysmal nocturnal hemoglobinuria, which I know is a mouthful, but these are rare diseases that are caused by dysregulation of the complement pathway, which unfortunately is not related to what we're dealing with here. And so I wouldn't expect this medicine to be effective for these diseases. I know someone was asking this question, and I know that Tony is also going to answer many of your other questions. And I want to also, as Tony mentioned, also iterate, I'm a nephrologist, an adult nephrologist, as well as an investigator. I spend almost all of my time doing research, but I would be happy to help you answer any questions or direct you to some of our clinical colleagues who can help you. The foremost clinician for these diseases in the United States is, of course, Tony Blyer, and John Sayer in the UK, and I think he's also here today. So these are great resources, these physicians, colleagues of mine, and friends. But I'm also happy to pitch in and help in any way I can, both for the science as well as for any questions about clinical matters that I can help with. Okay, so let me just, Paul, is it okay? Should I show some slides first to frame the conversation, or would you like to jump
Sure.
into questions? You're the
That's what
chair,
I would
so it's
do.
up to you.
Well, I think if you can go ahead and walk through some slides, and then we can make it very conversational afterwards. I've collected some
Awesome,
questions also from folks, and I
awesome.
think you'll find yourself on the hot seat. No,
Okay,
just
no,
kidding.
no problem. I'm happy to do what I can. So okay, so here's just a few slides to frame who we are and what we do at the Broad Institute. And we are, of course, very focused on the kind of research that will ultimately lead to therapies. These are my disclosures. It's really important for me to disclose specifically that I have been working on, we've licensed basically some of our discoveries out of the Broad Institute into a new biotech company. Richard calls this an affiliate of the Broad. It's not actually an affiliate, it's an independent VC-funded entity, and my only relationship to them is to be an advisor to the company. But of course, I care deeply about what they do in terms of progressing toward the clinic. So it was funded by several VC funds, and it remained in stealth mode. And it is now partnered with Alexion and AstraZeneca. So it's important for me to disclose this. And it's very much a journey that is still ongoing, and hopefully will result in a treatment that can make its way to the clinic. So my team, again, I'm a physician scientist with an emphasis on the scientist part. That's where I devote most of my time. There's a great need for us to make sense of, how do we take this great plethora of disease genes, so many disease genes that we have uncovered, and how can we make sense of how these misspellings in the DNA, if you will, what do they do inside our cells? How do they cause circuits to break inside our cells that ultimately lead to disease? This is the specific area that my lab specializes in. And of course, we try to use then these discoveries about cellular mechanisms to try to build specific therapies or candidate therapies for the clinic. This is a very common trajectory for many scientists. My colleague earlier, who also shared her mother's story with MUC1 kidney disease, sounds like she's doing very much the same kind of work. And this is really important work so that we can understand how we go from genetics, from deciphering the misspellings in the DNA, to ultimately developing therapies. And it's a very big field of inquiry across many different disease areas. And so this is exactly what my lab at the Broad Institute is focused on. And just to give you a sense of what we have done and where we hope to go, we are, if you will, bullish on the idea that we can get a therapy to the clinic because we have done it before. So earlier on, and this is work that is not related to MUC1 and UMOD, but earlier on in my lab, we worked on another set of mutations that activate or inactivate certain pathways in another part of the kidney, the filtering part of the kidney. And we focused on this target, this ion channel, and we built specific drugs or medicines that specifically work on this ion channel. And we were able to take this from discovery in my lab back in 2013, 2017, all the way to phase 2 clinical trials in patients in 2021, 22. And so this work has been completed, and this project is now with Bristol Myers Squibb, hoping to make it all the way. We're very hopeful for this, and the reason to believe that we can do this again is because we've done it before. So this is what I wanted to mention, that our group has been successful in the past to bring it all the way to patients in phase 2 clinical trials in patients. By the way, those patients responded beautifully to this drug. And so again, we're very hopeful that we can do it again. And so today we will be focusing on MUC1 and UMOD, which my lab has been working on. And of course, we're hoping that we can enter phase 1 in 2027. So we're farther behind, but making our way. And I just hope that I can convey to you that while this is a winding path, there is a way to get there, and it's been done before, and we hope to get there again. So, I'm trying to bring hope while also making, keeping it, realistic from the perspective that, it's definitely not a straightforward path. Okay, and just to briefly mention, I mentioned it yesterday, and the details don't matter, but where you see green, that is bad, that is the mutant MUC1 that is accumulating. These are human cells from a patient who has generously donated a piece of their kidney so we could develop cells. What Dr. Blyer just mentioned is a very valuable tool for us, like truly, truly enormously valuable. This is a mouse that has the same mutation as the patient's. And as you can see, there's a lot of green mutant protein in the kidney of this mouse. Well, this is the kidney of a mouse, I should say, a slice of a kidney of this mouse. And this is a human organoid, a mini kidney in a dish, if you will, that we developed in the lab. Again, patients generously donated some of their blood. We took blood cells and turned them into pluripotent stem cells. And from there, we developed organoids, meaning mini kidneys in a dish. And from there, we were able to test the organoids that have the mutation from patients that have the mutation. They developed a whole bunch of green, bad mutant protein, if you will, right? And now this shows you what the drug is doing. You can see that the green is very effectively going away from all of these different scenarios, telling us that we have a start for a therapy. This is not a therapy itself. This is just a tool that we are going to use to develop a real drug. And so that's what we're doing right now. But to give you hope that this is what's happening, and it's not just in mice, as is shown in the center panel, but it's also in patient cells and in mini human kidneys, if you will. So we're very hopeful that this will work in humans as well once we get to this. And the terminology doesn't matter too much, but if you want to know, this drug targets a specific mechanism that we discovered in our lab, and it's called TMED cargo receptors, and this is what we're working on. Think of them as cargo trucks that accumulate the mutant protein, the toxic protein, and they don't let it go. And in the presence of the drug, we can send the cargo trucks into the recycling facility of the cell called the lysosome, and that's how we're able to get rid of the mutant protein. So that's the general principle of how this drug works. And it also works in UMOD, and this is just showing you, yesterday there was a whole presentation about UMOD by one of the wonderful people in my group, Silvana Bazua Valenti, who worked on this project. But just to give you a little snippet to show that the mutant protein is here. These are the black lines that represent mutant protein in the mice that have this mutation, like the human mutation. And here's after the drug, the same, the siblings of these mice that have the mutation. There's a lot of reduction of the mutant protein in the presence of the drug, again, suggesting that working through these TMED cargo receptors, we're able to get the mutant protein to go away. So it works for UMOD and it works for MUC1, and it's a tool that we can now use as a starting point to build a suitable drug that can go into patients. And then the most interesting thing that we discovered, which I realize may not be of immediate interest to you, but it should be, and I will explain why, is that this mechanism that we uncovered in the kidney is not only operative in the kidney. It also affects patients who have families in which they have familial Alzheimer's disease. That means Alzheimer's runs in the family, so that people at my age would already have Alzheimer's disease and dementia. And so we can help those patients. It helps patients who have familial blindness. That means in every generation, half of the people in the family go blind from a mutation, another misspelling in the DNA. And so we can help those patients as well. And on, we are finding now that it may have an involvement in some diseases that run in families that affect the liver, the lung, and other organs. And the reason that I'm sharing this with you is because this is an opportunity, this nodal biological mechanism is an opportunity for this disease to become much more desirable by pharmaceutical companies to develop, because it is actually likely to help many more patients. From their perspective, they're happy because they can sell their drug to more patients. Of course, they are in the business of making treatments for patients. From my perspective, it is very meaningful because we can have an easier path to approval potentially, and also because I can help more patients, which is of course my mandate. I'm a physician scientist. This is what I trained for. And so the opportunity to help more people is both strategically helpful to your community. It is helpful to other patient communities. And of course, it's helpful to, everyone involved in the drug development process because we can simply help more people. So I wanted you to know that this is also something that we're interested in. And so this nodal biological mechanism is basically how we uncovered how we go from a misspelling in these genes to these TMED cargo receptors and how we can harness this understanding to build a treatment that will hopefully make its way to the clinic. And so the current status for the treatment, as I mentioned yesterday, we started out from the tool that I mentioned to you earlier. We built another molecule that we thought was good for the clinic. Last minute, there was some toxicity to that molecule that was not related to these TMEDs, but it was related to what we call off-target toxicity, meaning something else was affected by that small molecule. It was sneaking into something else and causing trouble. So we could not move that forward to the clinic. So now we're back to the drawing board and pushing a new possible candidate drug toward the clinic. And we'll see where that takes us. We're still sort of in the process of doing that. And so that's why we were initially planning to do clinical trials kind of now. And now it's been pushed by a couple of years because of this unfortunate complication, which is, however, extremely common in drug development. I wish it hadn't happened, but it did. We know how to get over it, but it is one of those like twists and turns on the road of developing a treatment that is not uncommon. And the final thought that I want to leave you with is actually that we are not sitting on our hands just because we have this, approach with the TMED cargo receptors, which would essentially be a pill. This is the first effort to get there. But drug development or developing treatments for diseases is like climbing a tall mountain. So we are not just taking one path. This is the one path that is still on its way. But if our goal is to prevent, cure, or completely manage all of these diseases for all of you, then we have to take alternative paths as well. We don't know which way will get us to the peak. Maybe all of them will, if we're lucky. So in the meantime, my group is also working on what we call next generation treatments, and this is ongoing work. So this is now inspired by the work that we did previously, but now we are working on things like, for anyone who understands or is interested in this, nucleic acid therapies, things like antisense oligonucleotides and siRNAs. These have never really been developed successfully for the kidney so far, but there are some ongoing efforts, and we're very encouraged, actually, that we're making our way here. So we are working on this. And then, of course, the big aspirational goal would be that we do a one and done cure. Go into the kidney and fix the misspelling forever so that no one has to worry about, transplant or dialysis or taking pills every day. That would be a cure. We are very far from that. That is the gene therapy approach that we're working on. We're very far from that. I just want you to hear me. We are working on it, but it's a long road. So this is not something that will come around the corner, but it might be suitable for the next generation. And I know that many of you, of course, deeply care about the next generation. I'm a parent too, so I totally understand. And so we are working on that, and we hope to be able to bring something forward. It might take us a decade of work on this, but we're determined, and we're going to try our best to make a gene therapy possible as well. And so I just want to skip through the next few slides and come to the most important slide, which is it takes truly a small village to make all of this work happen. So this is my team. And we all work very well together. I am, honored and lucky, frankly, to have such a talented team of scientists who are so very dedicated to the cause. And of course, an incredible team of collaborators, many of whom are on this call and you've heard from already, who all of us together are working in service of, finding therapies for these diseases. So I'll stop there, and I hope that we can have a conversation, have some questions, answers, and do the best I can to answer them.
Great.
Thank you.
Thank you very much, Doc. And one of the first questions I wanted to ask you is what we've seen over the past couple of years is FDA has changed in the area of diagnostic drug and device approval, where they're actually telling companies that are coming into FDA with trials and with drugs, looking at the product pipeline years in advance, that they are specifically looking for collaborations with patients and the company's use of patient insight data and lived experience data. And I was wondering if you could tell the audience how important that is from your aspect, looking long range in getting ready to go to FDA.
Critical, of course, Paul, as you well know, and I think, has as has been true for several other therapies that have been approved for kidney and of course for many other indications as well. Um, what we call patient-reported outcomes is one way to say this, or just as you say, the lived experience of the patient. What do patients care about? What are the symptoms that you want to address? I think these are all critical to all the regulatory agencies in the US as well as in Europe. And I think that this becomes a really important part of the approval process. So this is why, again, what I said in my sort of opening remarks that, when Richard and I were first talking about starting a foundation, I said, this is an effort that has to come not from me. I mean, I'm happy to help and be a part and a partner in the process as we are doing today, but that the patients have to be organized and, be ready in terms of partnering with, whichever entity, pharmaceutical company, startup company, whoever it is that brings a therapy forward to the FDA. The patient groups are critically important. And, participating in natural history studies, for example, that our colleague Dr. Blyer is conducting is critically important, what we call the registry. So the registry is one way to be counted. Then further, participating in a natural history study involves sharing a little bit more of your data than just the fact that you are part of the registry. And those are all very, very important steps that we have to take to prepare for clinical trials. So clinical trial readiness through the patient organization, and then partnership, as you say, all the way to approval. And the agency absolutely loves to hear from patients. And in fact, as you also said, if they don't hear, there's something wrong, right? Like they feel like maybe this is not the right path and that's not what the patients care about most. And so for any entity, a company, pharma company, startup company that goes to the FDA, patient engagement is absolutely critical. Patients as partners every step of the way. I think it's really important.
Okay, and then we'll get to Kathy has some questions from the audience, but I want to ask you one other thing so that folks listening understand how strategic this is. One of the other things that FDA will ask, and I say this as somebody who just got done 6 years of chairing the FDA's Patient Engagement Advisory Committee, they will often ask, what is the role of patients been in informed consent for clinical trials, and also what has been the level of engagement for return of results? And I'm
Mm-hmm.
wondering if you could kind of speculate on that or talk about that, because I know a lot of folks before they even get to the point of saying yes on trial, they want to know what is involved, who was involved with consent, and that type of thing.
Absolutely. And I think that those are all, again, this critical aspect of patients as partners at every step. As you also earlier said, like nothing should be happening for patients without the patients, right? That, it's critically important that, even when we're drafting a consent form, even when we are designing a clinical trial and we're saying, oh, we're going to, do this procedure on this date, and then 2 months later, we're going to do that, and then 3 months later, and then, patient input is really critical. Like they might say, oh, that's terribly inconvenient, or I don't think any of my fellow patients are going to sign up for that. But if you do it this way, then it would be more convenient and people would sign up. For example, just, this is not relevant to this disease, but for prior trials that I have been involved with our previous discoveries, you know, it used to be thought that you need to do a 24-hour collection of urine. Well, for most of us who work and have an active life, like imagine, like how do you collect your urine for 24 hours? You have to stay home, right? And kind of run to the bathroom every time, you need to collect your urine. Very impractical. And then it became apparent that you don't actually need to do that, that you just need to do, 2 or 3 early morning, collections that is just as good as the 24 hours. A lot of that input came from patients. And of course, if you stop to think about it, of course, it's terribly inconvenient and impractical, right? And so I'm just giving an example to say that patient input at every step is really, really important. And everybody looks for that. And I think, our colleagues in the pharmaceutical industry are very attuned to this as well. Of course, us, the academic investigators, are very attuned to this. And, partnering with patient organizations like the Rare Kidney Disease Foundation is going to be critical as we design our trials, figure out what endpoints we're going to use, the language in the informed consent, all these things, who gets to conduct the trials? Are they going to be, in XYZ center in the United States? Are we covering enough ground? What about patients who live in more rural areas?
Mhm.
All these things have to be taken into account. Are there ways in which we can be more adaptable, like send a nurse to the house instead of having the patient travel, I don't know, 100 miles to go to a center, right? So, and these days, I mean, we have computers, we have technology, it should be possible to utilize these tools. And I think, everybody's very interested in doing that. And I think for this audience, it's very important that we all hear that this is a patient-centric approach to clinical trials is the way to go.
Hats off to you, Doc. I know that Kathy has a couple of questions that she's gotten from the audience. Kathy, go ahead.
Sure.
Thank you, Dr. Greka. This is a two-part question. What are the chances that stem cells could be used to replace damaged tubule cells? And the second part is your lab doing any research in this area?
Yeah. Yeah, I mean, stem cells and cell replacement therapies have been attractive for a long time, and yet they haven't been as successful as one would think for a lot of diseases. So we are not currently working in cell replacement therapies for the kidney. There have been some efforts that have not been terribly successful. Part of the reason is because in the case of both MUC1 and UMOD, there are very specific kinds of kidney cells that are affected by the mutation and that unfortunately die off over time. And it is very hard to specifically replace those particular cells. And so I think, in the large context of what one would have to do, it's potentially harder than developing a gene therapy, which I already told you is like a very, very hard problem. So this is my scientific perspective on this, that I don't think that it's going to be as promising an approach as the other approaches that I just described earlier. So pills, nucleic acid therapies like ASOs and siRNAs, and then various gene therapies and gene editors that we're working on. So that's why we have not prioritized this area of work. But there are some efforts, and of course, I'm a scientist, so one should always leave room open for new things. And so it's possible that one day we will figure out exactly how to replace those very particular cells that we are missing. But at the present time, I'm not sure that we're very clever about doing that. So that's my best answer to that question.
Thank you. The next question is also two parts. Can MUC1 be traced back to a single population, or has the mutation occurred across multiple ancestors? And do we know how rare the first occurrence in the family is for these mutations?
Wow, yeah, that's an interesting question. And there's others here who, Greg, for example, from Cyprus has tackled this question and can tell you more about it as well. So others can chime in, but it's probably true that there were many independent initial mutations because truly there's no geographic limitation to this mutation. We find it in people everywhere, on every continent, as far as I can tell. There are founder effects. For example, in Cyprus, it's pretty clear that there was probably an initial ancestor, and then there has been, the island effect of sort of people intermarrying within a smaller geographic location often, means that there was an initial mutation and a common haplotype for all of those patients. That means a common stretch of mutated or misspelled DNA that they all share from an initial ancestor. So the best answer is probably that there were several spontaneous mutations. I think another reason to think that is because this area of the MUC1, the reason the MUC1 was so hard to find, this mutation, is because the mutation, the misspelling in the DNA, is hiding in a very dark corner of the human genome, if you will. And that's why it was so hard to find it, and it took a lot of special firepower by the Broad Institute to really get to that mutation. So that dark corner of the human genome is prone to mutation, if you will. And so it's likely that this is the reason why all of the mutations in MUC1 concentrate in that dark little corner, as we call it. The VNTR is the technical term, the variable number of tandem repeats region. So it's just prone to mutations, and that's probably why these mutations arose. And there's some evidence, we discussed this yesterday, that there could also be somatic mutations, which means new mutations that arise in an individual patient, not that they inherited from their mom or dad. So that's also a possibility that we are looking at in some cases. But the vast majority are inherited in an autosomal dominant pattern, as you have been hearing. So every generation in every family is affected, and there are probably multiple ancestors. Did I answer everything? I'm trying to think. Was there?
I think you did. I think Kathy's got one last question for you. Okay,
Very
Doc?
good.
Now we have time for, will pig kidneys be used in the future for kidney transplants?
Oh, yes. Not so much in the future, even in the present. This is really exciting work. So to back up for those who may not be familiar, as you probably know, there's a shortage of organs. There's not enough kidneys to go around for all the patients who need a kidney transplant. And so some of my colleagues, actually, there's several colleagues all around the United States and the world who have been working on this problem. A lot of it is based on what we call CRISPR technology. So CRISPR is a way to basically use scissors or tweezers to change genes, to cut them and snip them and paste in the correct kind of gene that you need. Or sometimes just to cut them out completely and eliminate them if they're bad genes, let's say. So CRISPR technology has received the Nobel Prize. There were two discoveries that happened simultaneously. One was at the University of Berkeley. They received the Nobel Prize, and the Broad Institute, my institute, also we discovered how to use CRISPR in human cells. So CRISPR has enabled something really pioneering, which is that you can generate kidneys in a pig that can have all the bad pig genes taken out, and like appropriate preparation of that pig kidney without any bad genes from the pig. So those are all gone. And now there's a functional pig kidney that is about the size of a human kidney, which is about the size of my fist. And now that kidney that came from a pig but has been prepared for a human, if you will, using CRISPR technology, can now be transplanted in a human. And in fact, that's happened a few times now. A couple of times, patients lived for a period of time, but then unfortunately they passed on. But now there's a patient who has received a kidney transplant who's still doing quite well. And these people are pioneers, and we will see how it goes, but there's a lot of promise for this. And I think that this could be an approach that can help us manage the shortage of kidneys for transplant. Of course, if our efforts are successful, we will never need transplants or dialysis. So everybody, all the scientists are working in their own sort of track to try to see what we can do to help patients. But this is incredible new technology, and there could be a time when pig kidneys will be more widely available. Right now, it's highly experimental. It looks promising. There's a lot of hurdles still, as with every new therapy. But thank you for the question. I think it's a wonderful new technology on the horizon, for sure.
Thank you very much, Dr. Greka. And at the end of the day, you're an optimist and you're a champion. And as I was saying at the top, and Rich knows this quite well, for everybody who's watching, understand it's often rare to find somebody who is a scientist, a doctor, and a passionate advocate all in one package. You're that package, and
Thank
this
you. You're very
foundation
kind.
and all of us as patients, we're very fortunate to have you in the arena alongside us.
Oh, thank you. I'm
Thank you.
fortunate to work with all of you. Thank you so much.